In December 2019, China notified the World Health Organization of several cases of human respiratory illness, a disease later named COVID-19. The virus causing this disease is known as severe acute respiratory syndrome coronavirus 2. The disease spreads through small droplets that are expelled from the nose or mouth when a person with COVID-19 coughs or exhales. Therefore, standing close to someone who is infected can put you at risk. These droplets can land on your hand and be transmitted through something as simple as a handshake, if afterwards you touch your eyes, nose, or mouth, the so-called T-zone. The virus is known to survive on different types of surfaces, so touching these contaminated surfaces and then touching your T-zone brings a high risk of infection. What we know so far: the coronavirus is spherical in shape, and its genetic material is encapsulated by different types of proteins. Some of the key structural ones are spike S protein, the most prominent feature of coronaviruses from where they get their name, then M, or membrane protein, and the so-called envelope protein.
Coronaviruses are a family of viruses that cause illness in humans and animals. 7 different types have been found in people, including those responsible for the SARS, MERS and COVID-19 epidemics. For replication to occur, the virus has to first enter the host cell and bind to receptors found on its surface. Of these surface receptors, ACE2 is what coronavirus's family members, such as SARS-CoV-2, use to gain access. ACE2 is also expressed on the surfaces of other cell types such as those of the lungs, thereby explaining why this virus is responsible for respiratory system infections. One way the virus enters the host cell occurs when the spike proteins bind to surface receptors, and the virus is engulfed by the host cell. The spike protein is then cleaved, leading to the merger of the viral and host membranes, and entry of the viral RNA into the cell. A more direct way is when the virus fuses with the cell membrane and depositing its genetic material directly in the host cell cytoplasm. This genetic material contains all the elements required for making more copies of the virus. The code for making the spike protein is here, as well as the instructions for producing the nucleocapsid and other structural proteins. Most of the RNA, however, codes for the replication machinery itself, and it is this machinery that is the first element produced. To translate its RNA into viral proteins, the virus uses its host cell's ribosomes. The initially produced viral proteins are referred to as polyproteins because their size is huge. And they will need to be cut into several smaller proteins, including the 2 proteases, which will do the cutting itself. Many of the newly produced proteins are then inserted into the various membranes inside the cell. Here, we can see the viral RNA being translated in the endoplasmic reticulum. In essence, the virus has hijacked the cell's machinery and created a new machine which can now multiply its viral genome. This machine is referred to as the replicase transcriptase complex. Replicase transcriptase complex uses the viral RNA, also known as plus RNA, to create a complementary copy called minus RNA. This minus RNA is then copied to make more plus RNA, which can be packaged into new viruses. The next elements created are the viral structural proteins. Here, replicase transcriptase complex first makes small minus RNA strands. This is in contrast to ribosomes directly translating the RNA as seen earlier. Different small strands, including the 4 types shown here, are in fact made in this manner. The replicase transcriptase complex then makes copies of the small minus RNA strands to produce small plus strands. These strands are now called subgenomic mRNA. And the host ribosomes can now translate these subgenomic mRNA into viral proteins. Among them are the nucleocapsid protein, the spike protein, and 2 additional viral proteins found in the viral envelope. Three of the structural proteins types shown here are made by ribosomes on the ER, where the proteins get incorporated into membranes, whereas the nucleocapsid protein is produced in the cytoplasm. This way the host cell is turned into a factory for making viral elements, the production of which occurs in networks of double membrane vesicles that form in these infected cells. While the nucleocapsid proteins code and protect the viral genome, the structural proteins move through the internal membranes of the cell, on their way towards the cell membrane. Their first stop is the Golgi intermediate compartment, where viral genomic RNA and membrane-embedded viral proteins merge to form viral particles. The virus hijacks the host cell's secretory machinery, moving newly created viruses through Golgi and finally to the cell membrane. Vesicles full of viruses fuse with this cell membrane, releasing new coronaviruses outside the host cell. This replication cycle repeats itself as these new viruses encounter other cells.